So we're going to start with a brief history of the development of recombinant DNA technology and of the context in which this technology evolved. Today, it's with synthetic biology and CRISPR-Cas9, genome editing, it can be easy to forget sometimes that we actually haven't known about DNA and how it works for very long. In 1944, Avery, MacLeod, and McCarthy were the first to demonstrate that DNA was the transforming principle, meaning DNA was the stuff that could cause one strain of pneumonia to turn it into another strain that was more virulent. And it wasn't until 1952 that fact was sort of locked in by Hershey and Chase's experiments. So not that long ago, we didn't even know that it was DNA as opposed to, for example, protein that was the stuff of heredity. In 1953, Watson and Crick, with the help of Rosalind Franklin's picture, demonstrated that DNA was in a double helical structure. So very recently, again, that we simply didn't have a good understanding of DNA itself. SV40, simian virus 40, was isolated in 1960. That will become important in just a minute. And in 1964, the Epstein-Barr virus was identified as the cause of Burkitt's lymphoma, so cancer. Prior to this, we knew that there were viruses that caused cancer in other animals. For example, we knew that there was a virus that caused cancer in chickens as early as the 1930s. But prior to 1964, we did not know that there were viruses that could cause cancer in mammals. Now, think about how terrifying that might be, right? This was a time when there weren't good treatments for cancer. Cancer was a death sentence. It was a terrifying disease to get. And to think that there was a virus that could give you this disease was particularly scary. 1972, EcoR1 was isolated from E. coli. And again, that will become important in a minute. It's also important to keep in mind that this was a time prior to many of the protections we currently have, not only regulatory protections, but also physical protections like fume hoods, etc. There was at this time a memorial lab at NIH that was dedicated to all those who had died in the conduct of their science. This was prior to pipette men. This was a time when people were mouth pipetting viruses and bacteria. And think about the people doing the research on a virus that caused cancer in humans. Think about how you would feel about mouth pipetting that organism. As I mentioned on the last slide, SV40, simian virus 40, was first isolated from monkeys in 1960, and it was carried and cultured in monkey kidney cells. These cells, or cells infected with SV40, caused tumors in hamsters. Okay, so we knew that this could cause tumors in hamsters. It was also the case that monkey kidney cells were used to cultivate poliovirus for the early vaccines. Polio, horrible disease. The vaccine was a tremendous boon, but it turns out that the rate of infection of these cells with SV40 is pretty high, which meant that some lots of the vaccine were actually contaminated with SV40. This was at the same time that we're discovering that there are viruses that can cause cancer in humans. And we've discovered that vaccine was actually contaminated with a virus that can cause cancer in hamsters. Terrifying, right? This was absolutely terrifying. So this, along with other many other instances that came to light at around this time of scientists behaving badly, basically, led to a fairly low opinion of scientists among many members of the public. So this was a time when, right at the same time, 1964, it was revealed that Dr. Chester Southam had been injecting live cancer cells into elderly patients at the Jewish Chronic Disease Hospital in Brooklyn without their consent. A few years later, in 1971, around 1971, the Willowbrook study came to light, in which children at the Willowbrook State School on Staten Island, New York, which was for kids with mental retardation, were intentionally infected with hepatitis in an attempt to track the development of the disease. Again, without sufficient consent. And then, of course, very shortly thereafter, there were the revelations about the Tuskegee syphilis study, which had been going on for decades, in which sharecroppers from Tuskegee, Alabama, who had syphilis, were kept from receiving adequate therapy, even after therapy was available for the disease. They were kept in the dark about that and not given therapy and allowed to live and suffer from syphilis. So this time in the 1960s and early 70s, not only was it a very sort of tumultuous time socially, in particular in the United States, but the scientific community was not as well respected, let's say, as we might hope it would be. So this is the context, the environment in which recombinant DNA technology comes onto the scene. So Paul Berg and colleagues at Stanford were interested in doing work with SV40 and had analogized the relationship between SV40 and eukaryotes to be similar to the interactions between bacteriophages and bacteria. And they started trying to get a bacterial gene into the SV40 genome. Janet Mertz was a new grad student and was interested in trying to replicate SV40 in bacteria. And she attended a workshop at Cold Spring Harbor in 1971 and started talking about these proposed experiments. And she met with some resistance. Following that workshop, Robert Pollack called Paul Berg and basically told him, don't use E. coli, right? Don't try to replicate SV40 in E. coli. E. coli lives in the human gut, and it's just too dangerous. And the cancer risk is unknown, but it's possible. And Pollack believed that no one should be able to do such a potentially dangerous experiment sort of in secret, only to present the completed work at a conference or in a paper, at which point the whole community, the whole scientific community and the broader society have to deal with the consequences. Following that, Paul Berg sought out additional opinions and indeed found that others of his colleagues were a little bit concerned about this research, and he paused those experiments at that time. So early papers started to come out that sort of were working their way toward the goal of replicating this virus in E. coli. And they had constructed, they had made the construct, but had not yet done the transfection. So let me back up a bit for the folks who aren't as familiar with recombinant DNA technology as others. So the basic idea here is that you're isolating DNA. It's like in your word processing program, cutting and pasting, but you're doing it with DNA. So you cut DNA at known cleavage sites, and then you use a ligase to glue the DNA back together in new combinations. And you introduce that recombined DNA back into an organism to replicate that new DNA. This was a huge advance over prior techniques, for example, breeding and crosses or random mutations. So, for example, irradiating your population and then looking for those mutants that had the trait you were interested in. With recombinant DNA, as with genome editing today, you can go in and directly make the change you're interested in. So DNA or restriction enzymes cut at palindromic sites. That is, sites in the DNA that read the same forward and backward, like a man, a plan, a canal, Panama. They create sticky ends, overhangs, as you see here. They can also create blunt ends. And then you can paste in your new DNA. Restriction enzymes are site-specific. They always cut at the same site in the DNA, that is the same stretch of bases in a particular order. The restriction sites are usually fairly short, 4 to 6 nucleotides. Most are palindromes, as I noted before. And we have made several hundred of these enzymes have been isolated from microorganisms. There are a number of different vectors that you can use to paste your new DNA into and use for replication purposes. It depends on the size of the piece of DNA that you are inserting and the host organism that you want to use to replicate the DNA. The vectors also have a range of sort of properties. The insertion sites can be, you can have distinct insertion sites, replication origin, selectable markers. So for example, you can have an antibiotic resistant gene so that you can plate out your organism on a plate that contains the antibiotic so that you know that anything that actually grows there has the DNA that you inserted because it also has the antibiotic resistance gene. You can make it dependent on amino acids, etc. And they can be used for a wide variety of purposes, including isolating a particular gene, production of a protein or RNA of interest, creation of recombinant organisms, etc., and potentially, of course, to correct human genetic mutations. And we do have already, with CRISPR-Cas9, some clinical trials that do just this. So at this point, enough has been accomplished in the science to sort of see the possibilities of recombinant DNA technology, but not really to judge the risks, right? They have done enough to know what might be possible, but not to sort of calculate the likelihood of bad outcomes, for example.